So make sure you're working these problems, right? Because again, I mean, I kind of expect you just to know the concepts and ideas, examples and whatnot in the lecture notes. And you use those to emphasize kind of what to study and what not to study in the book, so you should know this stuff. But the problems, you need to be able to do these problems relatively efficiently, right? You can't struggle with a pedigree problem for half an hour when there's, you know, a number of problems that you've got to work. So you have to do them somewhat efficiently. And that comes from practice, doing the problems. So be working on this. All right, so we're going to finish up a whole variety of topics related to quantitative genetics, although I have to say it starts to get pretty qualitative at some point. But a bunch of interesting ideas about how we think about nature and nurture, genes, environment, artificial selection, this kind of stuff. So the first thing that we're going to do is finish up an example in quantitative genetics having to do with these kind of outliers. And this example comes from this set of data on bean weights. And it was done by Johansson. This is one of several experiments that he did that are pretty well known. And the idea here is to address how the outliers' progeny are going to look like if you were to breed them together. So what you have here is a table with different weights of beans, not the most exciting thing, but still perfectly suitable for, you know, getting at fundamental genetics ideas. So 20, 30, 40, I assume they're grams or milligrams or something, but we're not even given the units here. So these have been divided up into categories, and then we're going to take adult plants from each of these categories, grow them up, cross them, and ask the question, what are the weight of beans that are going to be produced if we start with beans that were either very, very light or beans that were very, very heavy? Okay? And this experiment kind of reveals the interaction between genetics and the environment. And what you see is if you started with beans that were fairly light, the offspring tended towards the middle here, but they were a bit light themselves. So it's kind of a combination of, well, they're not stuck right at the original bean weight. They're moving towards the center. They're moving towards the mean. This is called regression to the mean. And yet they're not moving all the way over here. And that tends to suggest that there was some environmental component and some genetic component causing these beans to be ultra light. Their offspring actually tended to move towards the center again. So there may have been some non-genetic reasons for the weights being here. And likewise for the heavier beans, the idea is that you cross these type of plants. The beans tend to be a little bit heavier, but they also move towards the center of this phenotypic curve. So some of the reason that these beans were so heavy is a genetic reason, and some of it is an environmental reason. So this is kind of the idea of the experiment, and we have all these weights. We have some averages. They range from 40-something to 50-something, with an average about 47. So even if you're picking very light beans, the average tends back towards this kind of mean value. So a summary of this type of an experiment. Beans obviously vary quite a bit in how much they weigh. In general, the bean offspring tend to be similar to the bean parents, but there's a lot of variation. So light beans make plants that tend to produce a little bit lighter bean, but it moves to the center. Heavy beans tend to make plants that produce heavy beans again, but again, it moves to the center. So you don't stick way out in these extremes and you're not just fixed there. So the bean weight from very heavy to very light moves back towards the center. This is called regression to the mean. And an explanation for this is that the reason those beans might have been really light or really heavy was some environmental component. Okay, so what could that be? In this case, it could be, you know, whether it was at the edge of the field or in the middle of the field, or, you know, in a big pot or a small pot, however it was grown. Did it have a lot of light, little light? How was it oriented on what face of the plant? Things like this can have an environmental impact on the growth of things, and cause the beans to be very light or very heavy. But genetically, they tended to move back towards the center. Okay, so the point is there's both a genetic part and an environmental part that we have to consider. And we're going to spend some time in this lecture thinking about this partition between environment and genetics. And that leads us to our next topic. So this first part is very, very simple. Basically, you can think of the variance in a phenotype, some normal curve, as dividing itself up or having two different components. Partly it's genetically controlled, and partly it's going to be influenced by the environment, right? And we can call the genetic variance, let's say VG. So it's a variation in the phenotypic curve from left to right that's due to different allele combinations in organisms at one side or another side of that phenotypic curve. And there will be an environmental component which will smooth out the curve even more. So a certain amount of phenotype that you see in these curves is due to genes, and a certain amount is going to be due to the environment. And this gives us this really crude equation that's fancier than it needs to be maybe, but the entire phenotypic variation you see in a normal curve has these two components, an environmental component and a genetic component. And everybody already knows this, right? It's nature and nurture. We just divide variance in phenotype up into two parts, a gene-influenced part and an environment-influenced part. And these become very difficult to parse out sometimes. Genes and their effects are very complicated. How genes interact with the environment becomes extremely complicated. So we're simplifying this a great deal just to say that there's these two major components. Okay, so here are some pretend diagrams, theoretical cartoon diagrams, trying to give us some possible scenarios where two different genotypes are affected by environment in specific ways. So I'm not sure how useful this is. It's pretty self-explanatory, but the idea is we're measuring some trait here, which happens to be plant height, that's on this axis, and we have a temperature variation, some environmental variable that we're adjusting on this axis. And then for each of the curves, we're shown two genotypes. So I think what we need to think about here is that genotype 1 and genotype 2 are probably strains or variants of the same type of organism. It wouldn't make sense to compare a cabbage and an oak tree or something, right? So we're probably measuring strains, things that have different allele combinations but otherwise produce the same type of organism. So in that situation with different genotypes, the first example shows us a scenario where genotype 1 is fixed at a certain height, genotype 2 is fixed at a certain height, and these heights are genetically controlled. Genotype 1 is here, genotype 2 is here, and there doesn't seem to be any influence of the environment. So this is one kind of possible scenario. Maybe it's an extreme one, but that's okay. Okay, here's another one. Here the idea is strain G1 and strain G2 are both influenced by the environment in exactly the same manner. So that's fine. You wouldn't be able to distinguish G1 and G2 in an experiment like this. They respond identically. So here, the variation from this to this to this is purely an environmentally determined. This one, C, you have two genotypes which have their kind of intrinsic height differences, and both of them are also affected by the environment. So not only do they differ from one another because of the alleles they possess, they also show a environmental effect where they get taller with temperature. And the final one shows you two genotypes which are influenced by the environment. They're also different in terms of their genotype because at any given temperature they have a different height except for 25, but they're influenced by the environment in different directions. Okay? So these are, I mean, it's pretty, excuse me, self-evident what types of things you might draw, that you have two different genotypes consisting of a collection of alleles that vary from one organism to the other. You put them under various environmental conditions, and different things happen depending on how a particular genome responds to the environment. So these are just things kind of theoretical to think about this relationship between genes and the environment. Okay, so let's look at an example. This is a book example. It's one of the simpler examples in this chapter in the book. And we're going to try to partition some sort of phenotype into a genetic component and environmental component. And this is a kind of neat example. It involves mice, and we're simply going to weigh these mice. And we've got two cases. The first case, case 1, we're examining a population of inbred mice. And inbred mice are homozygous at every allele, right? They're clones of one another. You breed them against one another and you generate the exact same genetics in the offspring forever and ever. That's the nature of inbred mice. They're homozygous, okay? So they're essentially clones. So what we do is we measure the phenotypic variance in this population, let's just say in our pretend example, as being 0.3 square ounces for this particular set of inbred mice. And some notes, variance is squared. I'm not worried so terribly much about that because you see it used correctly and incorrectly in lots of different places. But the value 0.3 is measured experimentally. It's not something that you just say, oh, I know it's got to be 0.3. This has to be measured. So the mice are taken from the cages, they're put on a scale, you measure them, you do one of these distributions, and you apply some statistical measures, and you determine the variance. So this is measured experimentally, not something that's necessarily known. And we put this value into our really simple equation where we're trying to determine how much of the phenotypic variance is due to a genetic component, how much is due to an environmental component. Okay? And these things look more complicated than they are. It's really fairly simple here conceptually. So here's our fancy equation that says the entire range of phenotypes that we observe for some trait can be divided into that which is due to a genetic component, that which is due to environmental component. And this is a very simple idea. And so we put in what? 0.3 is the phenotypic variance that we measured. That equals VG plus VE. We set VG here as due to 0. And the reason is these mice are inbred, so we don't expect there to be any contribution from genetics to a variance in a phenotype. They're identical. They're clones of one another. They're identical twins or whatever, like that. So there is no genetic variation. So we're going to set that term here to 0, and that makes the entire variation that we see in this experiment all due to some environmental component. Okay, so a few notes about this. Case 1, population of inbred mice, homozygous at every allele. And we also suppose we account for big differences like male and female and young and old and stuff like that. Let's suppose we're not stupid and we're not measuring, very, very young mice and measuring very, very old mice or something like that. We're taking kind of uniform populations. Okay, so why is VG 0 here? I just mentioned it. The mice are inbred. We'll talk a little bit more about inbred organisms later in the course. They're pretty interesting, but inbred means they're genetically identical to one another. They have the same alleles of every gene present in every mouse. They're homozygous. They're clones of one another. There can be no genetic variation, at least in terms of sequence, for these inbred mice. So that's why we set that at 0. So the conclusion for case 1 is the phenotypic variance that we measured must all be due to some environmental component. So even if they're grown in an animal facility in cages and you control as much as possible, still there's going to be effects of lighting and night and day cycles and food and all this stuff that's going to slightly influence weight range. Okay, case 2. We have a population of wild mice. These are outbred. They have lots and lots and lots of different alleles from which to choose to make individuals, right? So they're very diverse genetically. And let's suppose that we take a population of these outbred mice, we put them in the laboratory, and we measure the phenotypic variance for weight just like we did before, and we get a value for the variation. Phenotypic variation is 0.54 square ounces, so it's just a number, it doesn't matter, it's just a number. So as before, this is measured experimentally, it's not something that you would know necessarily in advance. They're put on a scale, and we're going to say that these mice were grown under exactly the same environmental conditions that we grew the inbred mice in case 1. So we think then that there should be no difference in environment in case 1 and case 2. And we've already calculated the variance that's due to environment in case 1 in the situation where the mice were inbred and there was no genetic component. And that variance was 0.3. So we think we can use the variation due to the environment of 0.3, which we've already determined for the inbred mice. So we now have two values: the total variation we see in the population, the variation that we've calculated that's due to an environmental component, and we know we have a genetic component here because there's so many different alleles that are contributing to different individuals. And so we just plug it into our simple equation where we've divided phenotype into a nature and nurture kind of component, and we get 0.24 is the amount of variation that's going to be due to some genetic component. So this is, I mean, it's conceptually very simple. The book describes a lot more about this kind of stuff that I'm not going to go into. It gets extremely complicated. You have to worry about gene-environment interactions and all sorts of things in these types of experiments, but we're just making a kind of broad survey and saying, yeah, there's a genetic component to phenotypic variance, there's an environmental component to phenotypic variance, and in these really simple examples, we can actually do some calculation to divide it up. Okay, so we have crudely partitioned the phenotypic distribution as being partly due to genetic component and partly due to environmental component. The book covers all sorts of stuff: inheritance, dominance, epistasis, all these things, and it's far too complicated. We're just surveying this, so just follow. Make sure you follow the lecture notes as a kind of guide, particularly in this chapter, which kind of is sprawling. Okay, topic 5, heritability. So we would like to know, usually, for a given type of organism that maybe is useful for us, can we breed those organisms together and select them for some trait or another trait? But to get some idea about that, we have to understand what types of heritability concepts we're dealing with. So we mentioned before that total phenotypic variation has these two components: a variation that's due to genetics, variation that's due to environment. Right? So that is pretty straightforward. What we would like to do is get a sense of how much is due to a genetic component, so that if we bred animals together, we could change their look or character or behavior or something like that. Okay, so we would like to make predictions about the phenotypes of offspring of some population, knowing whether there's a genetic component to whatever we're measuring. And we would like to estimate whether artificial selection experiments would work or would fail when we bred organisms together. So that's kind of the use of these types of heritability values, making these types of predictions. Okay, and there's two broad concepts for heritability. One is called broad sense heritability, which has a variety of different ways to denote it, HB, there's other things as well. Basically, broad sense heritability refers to all the types of possible genetic contributions to phenotypic variance. It includes dominance and recessiveness and epistatic interactions and allele to allele kind of cross-chromosome interactions, all sorts of things. Anything that has a genetic component, we kind of put under this broad sense heritability. So it's everything genetic. It turns out this isn't always that useful in breeding experiments. We need a slightly more refined concept to think about whether we will succeed or fail, let's say, in artificial selection. But broad sense heritability is at least a concept that kind of encompasses everything genetic and says a certain amount of phenotypic variance is due to this genetic component. A lot similar to what we just looked at. Okay, so broad sense heritability is denoted as the fraction or proportion of phenotypic variation that can be attributed to genes in general compared to the total phenotypic variation. So it's some sort of proportion. That amount of variation due to genetics over that total variation. There's another concept that turns out to be more useful in estimating whether we will succeed in artificial selection experiments, and that is narrow sense heritability. And again, it's given different designations, which I'm not going to worry about very much. But what we're trying to do here is say that there are 10 or 100 or 1,000 or 10,000 genes in the genome, all of which are contributing a little bit to some characteristic, to some phenotypic curve. And we would like to know, at those 100 or 1,000 or 10,000 genes, for all the possible alleles that are there in a population, can we kind of move that, sway that a little bit along that phenotypic curve towards some direction that seems desirable? So narrow sense heritability is kind of restricted to this ocean of alleles in a genome that give rise to these complicated phenotypic traits. It tends to exclude things like these big binary switches like dominance and recessiveness. It excludes things where one gene kind of overrides or controls another. It excludes things where one allele on one chromosome and another different allele on another chromosome may interact to produce some effect, things like that. What we're really saying is let's forget about all those kind of big jump type things, and let's consider this entire ocean of alleles that come in all these varieties and are different in one individual versus another, and ask, in a population, all this background stuff, can we kind of move it one way or another on some phenotypic curve? So most phenotypes are not due to these single gene jumps like we've seen before. There's this vast set of underlying alleles that we have to work with, like clay, that we can mold in some direction or another direction. Okay, so that's kind of what we're dealing with in narrow sense heritability. And we would like to know kind of the proportion of some phenotypic trait that's due to this ocean of alleles that's possible in a given population. That would be our narrow sense heritability kind of idea. So it's the proportion of total phenotypic variation that can be attributed to all these kind of small underlying allelic effects across the entire genome to set an organism at one spot or another spot. So it's variation due to these additive effects over the total phenotypic variation that we see in a population. And it's these alleles that we're trying to mold when we do artificial selection experiments, right? If you did an artificial selection experiment with some massive dominant recessive trait, well, it takes you a generation and you're there, right? In one generation, you see the switch either go one way or the other way and you're kind of done. But the question is, aside from that big switch that you can see immediately, what about all the other alleles in the genome? What about those? Do they still contribute? Can you still kind of move things around? And that's what we're talking about with this additive genetic variation. Okay, so here's a table from the book. We'll just assume these values are true at some location at some time, so on and so forth. The book kind of belabors its discussion of some of these things, and it just goes on, and you're like, oh, what in the world? So again, try to follow the lecture and the level of the lecture when you look at this section of this chapter. But they have a lot to say. Page after page, they have a lot to say. So let's look at a few examples here. Heritability, and we're talking about kind of a sense of the genetic component towards some phenotype. The first one here is stature. And we saw a picture of this before, right? Where we saw father height versus son height. And there was some connection, right? And yet it wasn't perfect. So in this particular table, we're given some value of 0.65, which kind of makes sense if you recall what those graphs looked like before. Another one, I don't know why they picked these, but anyway, milkweed bugs, wing length in milkweed bugs, okay? 0.87, that's pretty high. That's a pretty good predictor, right? That's a phenotype which is strongly under genetic control. What about things that are not? Those are interesting. Pigs, Litter size, according to the table, 0.05. That means you think that you can select a pig that has a big litter, breed that pig, and generate offspring that also produce big litters, and you will fail. Right? That's kind of what this is saying. Litter size doesn't seem to be under strong genetic control. Probably it's lighting, diet, sunlight, lots of other components are playing into a litter size phenotype and not so much genetic. Another one, poultry, egg production to 72 weeks. So what is that, a year and something or other? Not very high heritability value. So if you selected chickens that produced a lot or a few eggs up to this time point, bred them together, and asked, will you continue to produce chickens of that type? The answer is no, not so much. It's not a very heritable value. So there's other things, environment, food, lighting, emotional interactions, hierarchies, social context, all this stuff, which is affecting things like this, and that's not so much under genetic control. So that's kind of the value of these types of heritability values. You get an idea whether you might be able to succeed with artificial selection experiments and so forth. Okay, so artificial selection, what is it? Well, let's kind of contrast natural selection and artificial selection. The idea of natural selection, obviously, is this idea that alleles get passed to offspring in a way that kind of represents the fitness or the success of reproduction of the organism. And in this way, allele frequencies change from generation to generation to generation. And if you think of populations as really just collections of alleles, then the frequency of alleles changes from generation to 1, 2, 3, 4, and so forth. And if you think of individuals as just being picked out, in other words, you randomly select alleles here, you select them here, you select them here. If the alleles are different here and here and here and here, then the organisms themselves are going to vary a little bit as well. So this is the idea of allele frequencies change through time. That's the natural selection idea. And if allele frequencies change, then the types of individuals that you can make from those alleles changes through time as well. Artificial selection is the idea that we deliberately try to remodel alleles in organisms by picking organisms of a particular type and breeding them together. So if you think of a phenotypic curve, a normal curve, you'd like to pick organisms at one side or the other side for some reason, and you want to know, if I pick them at one side, am I going to shift the phenotypic curve towards those organisms that I select? Okay, so we would like to move along some phenotypic measurement curve, lots of eggs, few eggs, whatever whatever's being selected for. And this is done by selective interbreeding of organisms that have the desired characteristic. And if the characteristic has a high heritability value, maybe you'll succeed. If it doesn't have a high heritability value, then you might be spinning your wheels. So usually the point of this is to make food production more efficient, but sometimes it's just for decoration or novelty. So, yeah, question?
As you would rebreed artificially, would the heritability go up typically? Like as you breed organisms and traits?
Yeah, so once you generate homozygosity in the alleles that are determining some trait, then that's it. Everything after that will always have that same characteristic. So you'll fix those things.
Will it slowly increase or will it just
Whether it slowly increases or quickly increases depends on kind of how heritable it is, how many genes there are that control a particular phenotype, how strongly they control that phenotype, things like that. So it's going to vary from in any given situation. Like for these cases here, it may vary strongly depending on how heritable it was in the first place. If it's very heritable, I mean, you're there already with 1 or 2 rounds of selection, right? But if it's not, you may have to really work at it, right? And even then, it may not work so well because it may be due to largely environmental components. So it's, Okay, so artificial selection. So here's a simple example of artificial selection in Drosophila, these fruit flies that we've looked at as a model for chromosome structure and non-disjunction and all that stuff. So this is kind of a nice little experiment. We're selecting flies for either liking to be in the light or not liking to be in the light. So that's positive phototaxis or negative phototaxis. And the picture is from your book. And so here we have some phototactic score. I don't know exactly what they're, how they're quantitatively measuring this, but they've obviously got some sort of a container with light at one side and no light at the other side, and maybe they've got a ruler or something, and they're selecting things at various positions along the length of this container. So we have a phototactic score, that might be position. And then we have number of generations that we're breeding the animals that we select, okay? So the idea is we start out with a phototactic score in the original population that happens to be around 8 something, okay, whatever it happens to be. So I guess they're kind of spread out. Some like the light and some don't. And then we're picking a group that's near the light, picking a group that's far away from the light, breeding those together and asking whether we can kind of continue to move and generate populations that tend to like the light, tend to not like the light. So with each generation of selecting for negative phototaxis, we're getting a stronger and stronger effect. So the population over here is a line of flies that really doesn't like the light very much. And the population that's selected for positive phototaxis is over here. And at the end of 15, 20 generations, we get a line of flies that genetically really does like the light. So we've artificially selected this population in 2 separate directions, which is kind of interesting, either against or for the light. So it works, right? So that's the first point. Does it work? Yeah, it works. If you start out with an outbred population with lots and lots of possible alleles, lots of genetic diversity, you can accumulate these alleles that cause a certain trait into individual organisms and breed those together. So this is kind of a purification process, right? You have a whole bunch of alleles at the beginning, but now you've kind of purified or selected for those alleles that cause this trait, or you've purified those alleles that cause this trait. So these flies and these flies are kind of filled with the alleles that determine positive and negative phototaxis. So it works. It's a kind of purification process. We reach a selection limit. That's another thing that we see. So the curve seems to go up and then it levels off here. And the curve seems to go down here and it levels off. And the question is, well, why do we reach a selection limit? And there's a couple of possible reasons. One is you only had so many alleles in the beginning population that caused negative or positive phototaxis, right? I mean, maybe there's 10 or 100 or 1,000 or something like this. And after a few rounds, a few generations of breeding, you've kind of found all those, right? You've got them all in this population, and there's no more genetic diversity that would allow you to select even more. You're kind of done, right? So that would be one reason why you would level off here and here. No more genetic variation was left. If you thought you could select for flies with even greater negative or positive phototaxis at this point, you'd have to bring in a kind of a new population of flies with a new set of alleles and see whether you could push the system even further. But with what you've got here, with whatever you started with, you may be at the kind of the limit. You found all the alleles and put them all in the individuals. Okay. Oh, and the other reason it levels off is as you select for one thing, you might also be selecting for some sort of a negative side effect. So it's possible that these flies become dysfunctional. Maybe they no longer breed efficiently, and so you just can't even do the experiments anymore. Maybe these guys, they just hide in the corner, right, and do nothing. It's possible. So as you select for something, usually there's some other things that go along with it, and it could be that they're behaviorally compromised, and so you just can't go any further as a consequence of that. So that's another possible reason for why you level off here and here. Okay, so we would like to know whether an artificial selection experiment is likely to be successful, whether there's in this ocean of genes a whole bunch of alleles that contribute to a particular phenotype, right? And can we we find those? Can we select for those alleles and put them into organisms and make strains that have stronger characteristics? And we have a fancy formula that tries to use narrow sense heritability as a kind of proportion or ratio to give us an idea whether we will succeed in this type of experiment or whether we will fail in this type of experiment. We'll use this narrow sense heritability value to try to get an idea what is happening in an artificial selection experiment. And we have this simple equation that has these three terms. R is the selection response, which is how good selection worked in the offspring compared to the original population. Right? So it either worked well or it didn't work well. So that's kind of our R value. The H is the narrow sense heritability. That is basically kind of a sense of the proportion of this particular phenotypic characteristic that has underlying genetic basis across all the thousands of genes in that organism. So if it's strongly genetically controlled, then we're more likely to be successful. If there's virtually no genetic control, then we're not likely to be successful. And S is the selection differential. This is kind of where the parents were, the selected organisms were on that phenotypic curve compared to the original average. Okay, so I know it's a bunch of words. We'll see some pictures that I think will make this more clear, hopefully. So here's the equation. It's not that hard, unless I put some pictures in a moment to kind of explain what's happening. So let's see, in other words, so in words, the outcome of an artificial selection experiment depends on what your chances were, that is, how much of that trait is due to some additive genetic component, and the point on the phenotypic distribution curve where you started, where you selected individuals in your artificial selection experiment. And hopefully these ideas are illustrated here. So I've got some phenotypic distributions in cartoon form here. So we have a population that, if we made measurements, gives us a distribution that looks like this. It doesn't really matter what it is. It's just some set of numbers of organisms on this axis and the measurement of the trait on this axis, like we've seen before. And so let's suppose we get some normal curve as we might expect for whatever this is, coloration, height, something like that. So this is our starting population. And I have then three possible outcomes of an artificial selection experiment. One is here, one is here, and one is here. So in our original population, we make all these measurements, and we determine that there's a mean phenotypic trait at this position, but the organisms spread out around the mean like this. And we're going to now select parents in this population at this point. So whatever this measurement is, we're going to select parents at or around this point here, indicated by the dashed line. So they're stronger for this trait over on this side. Okay? And we're going to ask the question, if we select parents here, what will happen when we breed those parents together? Okay? So a number of things could happen, and that's kind of what I'm trying to indicate here, and here, and here. So what about this one? In this one, what happened is the F1 produced by breeding the parents we selected at this point all went back to the original mean right here. Right? Don't worry so much about the height of the curve, I just made them list something. But the point is, we selected parents here, but everybody in the offspring went back to the original. That would suggest there wasn't really much genetic component into why these animals here looked like this. Right? Maybe there was environmental effects causing them to be right here, but when you selected them, bred them, they went right back to the original population. Second one, here, let's suppose that we select the parents here with this at this point on the phenotypic curve, and the F1s look like this. In this case, the parents that we selected here gave rise to offspring that were very narrowly centered about at the point of the parents themselves. That suggests that the reason these parents were here was because this phenotype was under very strong genetic control at this point. So you take these, you breed them together, and you get exactly that back again. That would indicate a very high genetic contribution to the traits along this curve. So both of these are kind of the extremes, right? In the first case, you have seemingly almost no genetic control, so your experiment will never work. You'll never, in this case, you'll never be able to select animals and move yourself in one direction or another because it doesn't seem like you have any genetic contribution. In this case, you're done in a generation or so. It's almost purely under genetic control, which is also fairly unlikely. Okay? And the last one is kind of an intermediate example where the idea is the original population was here, the parents we selected were here, and the F1 is somewhere in between. So we didn't go all the way over. It wasn't solely under genetic control. We didn't kind of revert back to the original population, which would suggest it's not under any genetic control. We're somewhere in between. So we succeeded, we improved. We moved a population of organisms along some phenotypic curve, so now our new population is somewhere in between here, and then has some supposed agricultural advantage. So this is kind of the idea of what we're thinking about in terms of these artificial selection and the success or lack of success depending on the additive genetic neurosense heritability component. Okay, there's an example in the book where we look at fruit flies again, and we're going to be given some simple numbers, and we're going to try to determine some of these values with this R and S and neurosense heritability. So we have a population of fruit flies with a mean body size of 1.3 milligrams. So fine, it is what it is. Population of flies, we measure them, and we get some phenotypic curve where the average is 1.3. Half the flies, we're just going to take them, no matter what their weight, and we're going to breed them together to generate F1 offspring, and that will be kind of our control. That will show us that there isn't any big environmental effect in our experiment itself. If we take a population of flies with a normal distribution and we breed them all together, we probably are going to get the same thing out again in the F1. So half the population is bred to kind of prove that point. The other half, what we're going to do is we're going to select flies that have a body weight of around 3 milligrams. So that's basically what we're doing here, is we say this is, let's see, this is 1.3, we're selecting at 3 for the parents. So that's what we're doing in the experiment. The other half, we select flies with a mean body size of 3, we breed them together, and we ask what's the weight of the offspring F1 flies. In this case, we measure the weights of the F1s, and they have a mean body size of 2. So this matches almost exactly what has happened here. If this is 1.3, original population, this is 3, the selected animals, the F1s ended up somewhere in between at about 2 in this example. Okay, so our little formula here is this selection response, that is how good it worked, depends on the proportion that was due to some additive genetic component, the neurosense heritability, times kind of the point at which we started, the place where we started picking the parents along the curve. So selection response is 2 milligrams minus 1.3. That's how far we went away from the average. So that's 0.7. The selection differential, that's kind of the parents that we picked, they were at 3 compared to 1.3 in the population, so that's 1.7. So in this simple example, we divide this by this, and we determine a narrow sense heritability proportion of about 0.41. So this is kind of the level of problem that I think is suitable to do. It's simple arithmetic. You have to understand kind of where you're going, what you're trying to do here, but we don't get into all the detail that the book tries to go over in terms of words and all these exceptions and exclusions and so on and so forth. Yes, question?
So what was your problem for the offspring controls?
So, I mean, in our example, the way we're looking at it here, environment must be a component in this, which would be the other difference, right? So there's a fraction of the phenotype that's due to genetics, which we've tried to determine here. Whatever is not due to genetics, we lump into something else. It's called environment, right? But you can set this problem up in different ways. You might be given this or this or this or something like that, and it's a pretty straightforward formula. You just have to understand kind of what you're doing, where you're going with these things. Okay, so some examples, these are more fun. Examples of natural and artificial selection. So here's the first example, dogs. Right, so we have a whole bunch of dogs pictures here, and you could vote. Okay, so you have a choice. Okay, do you take this dog, or do you get this dog? Which one are you going to pick? This one, right? The puppy. Anyway, they all look different, right? What's the difference? They have different types of hair. They've got different sizes. They've got different colorations, different face structures, things like this. By and large, these things have been accomplished over the last 15,000 years or so, right? So this shows the results of deliberate artificial selection on the dog genome to generate all these varieties. It's really, really incredible. And I should say that you've got two things at play here. One is artificial selection, where you're kind of molding, like clay, all that ocean of background alleles towards one direction or towards another direction to generate some desired outcome. That's true. But the other thing you've got here is spontaneous mutations that may suddenly arise, giving you some sort of unique or desirable phenotype. These are things that are unpredictable, unexpected, not really part of the artificial selection process itself, just accidents in genes that control things like fur color, size, stuff like that. So there's a combination of things that are happening to generate this type of variety. This slide just is to show you that people are actually interested in trying to understand what those genes are. This is a paper from a number of years ago, tracking footprints of artificial selection in the dog genome. People will compare the genomes of all these different breeds, try to determine what are the differences, what genes are involved, and it turns out it's not that many genes. If there's 20,000 genes, maybe 100 or so can explain an awful lot of variation. Genes that affect size, that might be growth hormone type things or receptors. Genes that affect fur quality, well, we've seen some examples in mice where we can generate agouti, black, brown, yellow, and that isn't that complicated in the genetic system, but you get very different coloration patterns, right? So not so many genes. Color, fur quality would be, I guess, length of fur, things like that. You can imagine that it doesn't take too many different genes and you can cause a big variation in the output. Anyway, you can read the abstract here and maybe the introduction and just get a sense of what people look at when they do these types of studies on artificially selected animals like dogs. Okay, so let's think about a problem, kind of interesting problem. Feral dogs. One definition of feral dogs is the dogs that are produced if domesticated dogs are released and breed together, and then these dogs kind of are out in the wild and try to hunt for themselves and all that kind of stuff. Okay? So let's suppose we start with a population of primordial dogs, and we're going to breed these dogs or select these dogs or watch mutations happen in these dogs and kind of see what happens. So you breed them, mutations happen, and you generate a fancy breed A, fancy breed B, fancy breed C. And let's just suppose in this kind of simple artificial example that breed A is the result of a mutation in the A gene, and breed B is the result of a mutation in B, and breed C is the result of a mutation in the C gene. I mean, this is very simplistic, but you can kind of get the idea. It's plausible. So now the question is, we have these three breeds and we release them into the wild, they breed together, what's going to happen? Well, you're probably going to lose the A, you're going to lose the B, you're going to lose the C, and what you get back is maybe a little bit more of your primordial dog, your feral dog. And sometimes this is what people actually see in situations where dogs are released into the environment. The dogs are of different types, but you tend to lose some of these alleles that cause them to move along some lineage to make a specialized dog. They tend to revert back to this primordial or feral type of genetics. And these things are what? They're more like an Australian dog or a dingo, or they tend to be kind of short-haired, tan color, medium weight, pretty thin. There's a particular look to these types of animals. Okay, just something to think about. What about this? This is a totally different situation. If you look at this picture, you'll say there's no difference here than in the dog situation. the variation you see here doesn't seem to be any more or any less than what we saw with the dogs. But this is a totally different phenomenon. This is kind of natural selection over millions of years, not artificial selection over 10 or 15,000 years. So we get these huge differences in look, but these differences are accompanied by huge changes in the genome, right? I mean, they're still birds, right? But there's big differences in alleles, there's big differences in mating behavior, feeding behavior. There can be chromosomal differences. The number of chromosomes doesn't have to be the same. We generally think of domesticated dogs as being able to be bred with one another. But in this case, you have a much greater evolutionary scale or difference for birds than you do with dogs. So I think it's interesting because if you look at it, you say, this doesn't have to be any different than what we saw with the dogs, but in fact, there's a huge difference in the kind of background or origin of these. Some other pictures of artificial selection in pigeons. So this one, I don't know, sorry. This one looks like it's got a comb, almost like a rose comb type of a thing. And these are probably due to spontaneous mutations as well as artificial selection. So here's one. This one is, what, confidence? Is that this one might be color, I don't know. Does anybody, a pigeon person, knows that's unusual? Because it just looks like a pigeon to me. This one's got a mutation, obviously, that seems to have put feathers on the legs, so that's pretty unique, right? So then you've got here, again, what you've got are, you know, spontaneous mutations that people observed when they did breeding experiments, and you have, you know, deliberate artificial selection going on at the same time. Okay, so some examples. Another example, artificial selection in plants. There's lots of these type of examples, like in wheat, right? The accumulation of multiple genomes in wheat, which is a kind of characteristic of what's grown today. Here's another example. The idea is wild mustard gives rise to all of these different kind of food plants that you can now buy across the street in Sprouts and so forth. So artificial selection by people has generated all these kind of unusual characteristics. Mutations happen. People took advantage of those, bred them further, and you get all these things. Goldfish. This is more decoration. And again, there's a combination of, you know, artificial selection and breeding of these things in tanks year after year after year, and there are the sudden appearance of mutations. So who's the fairest of them all? This one's pretty bad. Anyway, so this is more decoration. Doesn't have really any, well, not much agricultural. And I got a picture of this guy. So again, due to artificial selection, it doesn't look like it could run very far if a lion was after it, but it did get a piece of jewelry, and it did manage to make its way into a painting. So its alleles were worth something, right? Somebody thought this is nice enough to make a picture of it, and then I show it in class. Okay. All right, the last few things. Correlation between two traits. How might two traits be correlated? So we're interested in this because one phenotype often varies with another phenotype. And when that happens, we often wonder whether the same genes are causing both phenotypes. So sometimes it tells us something fundamental and mechanistic. So we're interested in correlated traits. So it may be that genes that determine phenotype X also determine phenotype Y. That's possible, and that would tell us something about how development is proceeding, and that's important. Okay? So this is one reason why two traits might be correlated with one another. Another reason is linkage, and we haven't talked about linkage. We'll talk about it after the first exam when we discuss recombination. But if you have an allele of a gene at some location on a chromosome, and really close by you have another allele of another gene, and the first one controls trait X, and the second one controls trait Y, then whenever you see X, you see Y also. So they seem to be correlated. And they'll always go together because they're so close on the chromosome. So this is a kind of unique situation where two different traits can move with one another because the location of the genes along the length of the chromosome. So again, we'll look at this a little bit later. And chance. This is always the big concern in science. Are things correlated in some meaningful way? Are they caused by one another? Do they have the same underlying mechanism? Or is it just chance? So these are the reasons why we're interested in correlations between these phenotypic curves, X and Y. And I've got an example from a guy who links these types of things together to try to illustrate correlation and causation. So here we have a graph showing you that over a number of years, the letters in the winning word of the Scripps National Spelling Bee has correlated pretty well with the number of people killed by venomous spiders. And if you look at his website, there's a whole bunch of these things that he's kind of tabulated. And it's pretty amusing, but what he's trying to do is basically say, be careful about assuming causation, an underlying common gene that controls two phenotypes, for instance, as opposed to just some sort of chance correlation. So we should ask questions about this. My question is, how are the spiders doing this? It's amazing. Okay, so some examples of genetic correlations. Again, another table with lots of data from the book, but in chickens, egg weight, egg production, kind of makes sense. The idea is if egg weight goes up, there's another trait, egg production, which is falling. Okay, so those are negatively correlated with one another. And so they may have the same, genetic basis, or they may not, but they're correlations that maybe deserve investigation. Another one, probably more obvious, body weight and egg weight. As the chicken gets bigger, the eggs tend to get bigger. Okay? So I guess this one's point 4.2. I think a question would almost be, well, why isn't it even stronger than this? But it's not. So there must be a variety of things that are controlling these types of traits and their correlations. And there's a bunch of other ones with these peculiar examples, but those are a couple. Okay, so last few slides. I want to make a note about quantitative genetics and why we are looking at it. And the idea is, of course, that most phenotypes aren't due to one gene or two genes. You can't make, as I said before, things like elbows and knees and feet and all that with just one gene. As in Mendelian examples, you've got to have thousands upon thousands of genes all acting together to generate these types of complex structures. And in the news, you often hear about things like height or IQ or predisposition to cancers or similarities or differences between monozygotic and dizygotic twins and all this kind of stuff, autism, asthma, diabetes, whatever, all these things. The question is, are these single gene type of traits where we know one gene and it's kind of a binary yes or no? And for the most part, a lot of these things aren't like that. They don't fit that original Mendelian model. They're complex genetic diseases that have lots and lots of possible underlying influences that tend to set people at one position or another position along some phenotypic curve. And and the last point is these are hard problems. These are very, very difficult problems because there's so many different genes. It's hard to get populations. There's a lot of variety. There's environmental influences and all that. But the idea is kind of listed here, and that is for quantitative type genetic disorders, which are due to kind of the collection of alleles in one person or another person, the idea is that for those types of things, predisposition to particular disorders isn't as simple as yes, you have the allele or yes, you don't. It's more like this. So individuals with a collection of alleles can be placed anywhere along some kind of phenotypic liability curve. Some people have a collection of alleles that places them far away from one of these complex disorders over here. Other people are more in the center, and other people may be across some threshold. And if there's a genetic component to some of these, and it's lots and lots of genes, it just so happens some individuals get a collection of alleles that tend to put them over here towards a more liable spot on these phenotypic curves, and other people are over here. This is how we think about these complex disorders, and it's very, very difficult to figure out what this set of 5 or 10 or 50 or 100 types of things might be, and to kind of parse that out from all these environmental influences that also affect these things and push people towards a liability for complex disorders. Okay, but anyway, this is the way we think about complex disorders and the underlying genes that cause them. And the last thing, we'll stop early today, I guess, is I want to make a note about this chapter because this chapter is very difficult. And the note here is basically saying that presenting quantitative genetics in a single chapter in one of these genetics books is a very difficult thing to do. It either comes across as way too difficult or way too fuzzy, and I think the book does both of those things. It's lots of equations that we're not going to worry about, and other sections are just paragraph after paragraph, and your head spins, and you think, well, what's said here? So try to follow along with the lecture. The lecture presents the basic concepts, environment, genetics, narrow sense, a few simple equations. You ought to be able to do those types of things. So use the lecture as the guide for this chapter. I know that the book problems in this chapter are sometimes pretty easy, but sometimes are just absurdly difficult. And also they cover a lot of statistical things that I just care a little bit less about because we're a genetics class and not a statistics class. So I've got some things we're not doing. We're not calculating correlation or covariation. We're not making lines based on regression analysis, which I know is, they like to do that in the book. We're not covering quantitative trait loci, and there's a long discussion about dividing phenotypic variance into all these different components. All I did was genetics and environment. The book does a lot more, and that's all fine and good, but for us, we have a lot to cover, and so I've simplified it. Discussion of heritability in the book is confusing. They say things are heritable, and then they say things are not. They kind of go back and forth. It depends on time and place, and et cetera, et cetera. So, read it maybe, but use the lecture as kind of the guide. In terms of calculations or number types of things, you should be able to understand the stuff that I've discussed in lecture. Simple statistical measures of a normal curve are also fine. That's pretty straightforward. And some of these things that I went over with examples. If you're given a few variables, you know the equation that you could kind of plug and chug and get out the other variables, you know what you're talking about. So things like this that I mentioned in lecture. So hopefully that's helpful. I realize this chapter is very complicated, but hopefully that's helpful. Okay, so we'll stop early today. On Thursday, we'll talk about gene function. It's early short as well. So I'll see you then. Make sure you work on these problems for the exam on Tuesday.
I can take it from you.
She has her own.
Nerve of her getting her own backpack with the sister bottle. Is it younger or older? Younger.
She said you take now.
Okay.